So our next video is basically going to be the continuation where we left off. I have a question here, which is RNA splicing. Now, do RNA splicing occurs in prokaryote? Well, it would be better if I were to ask this again real time, but no mind, okay, this is something for you to ponder. Probably when we meet next, okay, we can discuss on it. Now, what I would like to continue on is actually on the translation. So like I said, since this is actually not really the focus of this course, so I'm just going to explain about it briefly because in actual the process is very complex. So for translation, it is to produce protein and okay, it will decode portion of mRNA in the form of 3 nucleotide or codons for one amino acid. So try to refresh back your mind on the translation for some of you. So the mRNA would comprise of ORF or open reading frame, and this would specify a single protein. So process of course starts similarly at 5' end with a start codon. There's only one start codon, AUG, rarely GUG or UUG. Okay? Because sometimes if you read some journals or go to certain websites, they say that GUG or UUG could start it, but it's very rare, in a very rare condition could it start. So it specifies first incorporated amino acid, and of course again it will determine the possible reading frame. So it will terminate when it encounters a stop codons, and there are 3 stop codons available, either UAG, UGA, or UAA. Alright, so there are other types of RNA for translation aside from mRNA. mRNA is the one that produced after transcription, but earlier on, okay, if you remember, okay, during the introduction of gene expression, there are lots of RNA that is going to be produced. The first one is tRNA or transfer RNA, which is going to be crucial, okay, to translate nucleic acid into amino acid, because it requires 20 enzymes to attach appropriate 20 amino acid to a tRNA, and the tRNA with the correct attachment of amino acid will be referred to as aminoacyl tRNA that will bring it to the strand of peptide sequences or polypeptide of protein that's going to be translated. So second one is rRNA or ribosomal RNA. We know that ribosome is actually a big part of the gene expression. So ribosomal RNA is used to make ribosomes for protein synthesis. So the ribosomes provide location for the translation. It's kind of like a house or a machinery, if you may say it. Okay, so it has 70S ribosomes, which comprise of 50S subunit and 30S subunit. And don't ask me why it's not 80, why it's 70. That's what it's called. Okay, but it comprises of 50S subunit and 30S subunit. So this is the 50. Okay, so this is the 30. So it has 3 pockets, as you can see over here. So the A side pocket, okay, whereby the amino acid will be basically attached to it. Okay, the P is for the peptide, the growing one, and E is for the acid, for the growing chain of peptide, to be released. So overall process of replication up until transcription and also translation is, as you can see, over here. So it involves 4 steps. So similarly, step 1 is initiation, whereby the tsRNA recognizes Shine-Dalgamol sequence at the mRNA strand and anti-Shine-Dalgamol sequence of the 16S rRNA. Will bind to it. So this is more or less how it will go. So ribosome will identify the correct translation site via initiation factors, IF1, 2, and 3. So the FMET tRNA or formylmethionine tRNA, the ones that brings AUG or the start codon, the only one is going to be needed. Whereby what happens is that the sludge subunit which is the 50S rRNA, the ribosomes clamps to the top of all components where the tRNA is situated at the P site. So first you have the small, and then you have the FMET, and then you have the large ribosome enclosing it. Alright. So what happens then the tRNA enters the P site. Okay, so if you don't remember, the P site is actually the one in the middle. Okay, and binds to AUG and IF2, okay, the initiation factor 2. Large ribosomal subunit joins the complex, okay, and this is the processes. So it will assemble the complex initiation, and whereby, okay, resulting in 70S initiation complex. So afterwards, okay, transcript could be translated into protein based on the codon sequences. So this genetic code table is just to refresh back on some of your mind. And for those of you who haven't learned about this, then well, I'm not going to go into details, but you can ask your friends or you can learn more, okay, in some books, or you can read on about it again via Google. So we only have 20 amino acids. Let me just say this briefly. Generally, we have 20 amino acids, okay, but we actually have 22 amino acids now that has been recognized. So we're not going to go into details for that. So, but the problem is we only have 4 nucleotides, right? Okay? And U is basically because we now have mRNA instead of T. So we only have UCAG. We have 20 amino acids. How do you divide that? Okay, it has been divided for you. Okay, so now when you combine, okay, all this, okay, it will produce 64 codons. Okay, you have 2 minus 4, 1 for the start, AUG, over here, methionine. Okay, then the other 3 is for the stop. Okay, stop. Okay, UAA, UAG, UGA. So the rest of them make up the 20 amino acids, including the methionine. We have phenylalanine here, leucine, isoleucine, valine, and so forth. How do they divide that? It's actually a very complex process. There is a history to that, okay, but I'm not going to tell you in this course. So the next step is elongation, similar to the transcription. So entry of the second charge tRNA, which will bind to the GTP-EF-TU into the A site. Okay, so hydrolysis of the GTP occurs, and the EF elongation factor, okay, TU-GTP, okay, is going to be released, as you can see over here. Okay, it's going to be released, and the peptide bond made between the two amino acids, okay, will cause deacylation of tRNA at the P site. So when deacylation occurs, okay, which means that, okay, it will move along over here. So the third step, okay, not termination yet. Okay, remember it has 4 steps. So the third one is translocation, which means that it moves. Okay, so shifting of growing amino acid chain. So the ribosome translocate along the mRNA strand by shifting empty tRNA to E site, and the growing AA tRNA to the P site. So translocation readies the A site for the next charge tRNA. So remember, this is E, this is P, this is A site. Okay, so process continues on and on until stop codon is reached. Okay, so your growing protein is being here. So this is the elongation. So we have E, P, and A. So the new one, okay, will be added here. Okay, the ones that has been made, okay, stays there. So if there's a new one, okay, it will stay here. But if there's another one, okay, so it will be shifted here, and then later on being released. Alright, so the last step, okay, the ones that everyone will be waiting for, is the termination, okay, whereby we have to terminate the protein. Okay, so it has to encounter stop codon, which means that no tRNA will be supplied. Why no tRNA? Because it's not charged. So peptide chain, okay, will be released, okay, from the ribosome by the release factor. So dissociation of ribosome from mRNA will take place, and of course polypeptide, okay, the peptide chain, the protein chain, okay, will be released. So the protein is produced from the N terminus to the C terminus, sorry, not TP, C terminus. So we don't call it 5' to 3'. The 5' is the N terminus, the 3' is the C terminus. So the N to the C terminus, N for the amino, C for the carboxyl. Alright, so basically, okay, what we will see is that, okay, how elongation continues, okay, and the process repeats until reaching a stop codon, and how it is terminated once it reaches the stop codon. No new charge amino acid will be supplied, so the process just stops. Now, how do the ribosome know which amino acid to add? Okay, so the question is that the answer is here in the diagram. So tRNA molecule carries specific amino acid, and they can each carry only one type of amino acid. So an N-type codon, okay, on the tRNA is complementary to the mRNA codon over here. So that's how it knows, okay, which amino acid to add. So just to test, okay, and this is for the fun of it, can you find the sentences? Can you read them? Can anyone read it? How about now? Okay, so the first one, okay, the man was too old for the war. Second, the boy had one red car and one old hat. Third one, the car was red and too old for her new job. So all of them have three alphabets, okay, forming one word. So this is representative of, okay, nucleotides and the codon for each amino acid. So this says what occurs during the translation process. So it has to find the methionine, the start codon first. And in this case, okay, a representative of methionine is the. So you have N, 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 which means that it stops. Okay? So what do they have in common? They have the, which is the start codon. They have the N, which is the stop codon. All of them basically are encoded by 3 nucleotides to represent one amino acid. Okay, so there you have it. Okay. So it's all in your notes as well. Now, how is this like the genetic code? Okay. So, of course, like I said, okay, it represents, okay, nucleotides with the codon sequences. So there are many words and phrases repeated or reused, okay, and we know that there are only 64 different combinations, but infinite polypeptide, which means that the combinations are endless, okay, or infinity. So it means that many are reused, which is quite common as well. But there is one big difference. The genetic code has only 4 letters. So how we do that? We have known that it is a combination of 4 times 4 to produce 64 codes. Okay, as you can see over here, the genetic code. Alright, so a summary of the processes in gene expression, okay, either the transcription or the translation. I'm including the post-transcription and post-translational as well. So for transcription, we know that this is the powerhouse of gene expression. It regulates which genes are transcribed, okay, from DNA to mRNA, or controls the rate at which transcription occurs. Faster rate, higher rate, okay, the quantitative of it. In terms of post-transcriptional, which we are going to look at in detail, especially for eukaryote, the mRNA molecules undergo changes in the nucleus before translation occurs. Like I said, okay, it happens mostly for eukaryotes. So introns are removed and exons are spliced together, and there are variations whereby you can produce different kinds of mRNA and even proteins from it. So once your mRNA is being translated, so translation controls how often and how rapidly mRNA transcripts will be translated into protein. So it will affect the length of time it takes for mRNA to be activated as well, and also the speed at which the enzyme will destroy your mRNA. So that is what happens to your mRNA after translation occurs. It will be destroyed, just in case anyone wondering about that. And the last part, okay, which is actually a control, okay, for gene expression and also protein expression as well, post-translational. So before many proteins become functional, and similar goes, okay, to mRNA, the transcripts or the gene. Some of them may be functional, some of them may not be functional. So protein, before it could be functional, they must pass through the cell membrane. And it's actually a very complex process. So a number of control mechanisms affect the rate at which a protein becomes active and the time that it remains functional, including the addition of various chemical groups. Right, so there you have it. I hope you have enjoyed this, and well, we shall see in the next time.